stop applying your face moisturizer to your chest every morning as if that alone is enough to preserve the skin there. That habit might feel responsible, even complete, because everything you've been told about skincare reinforces the same idea. Keep the skin hydrated, prevent dryness, and aging will slow down. But that assumption is incomplete, and the evidence is literally visible on the skin of your décolletage. The issue is not that you are doing nothing. The issue is that what you are doing never reaches the deeper layers where structural aging actually begins. The wrinkles forming across your chest are not happening on the surface alone. They are forming in the dermal structure beneath, where collagen fibers weaken, elastin networks break down, and fibroblast activity gradually slows with time. No standard moisturizer sitting on the outer layer of the skin can reach that depth in any meaningful way. It may soften the surface temporarily, but it does not rebuild the internal architecture of the skin. And that distinction is exactly where most conventional skincare approaches fail. What I am about to explain is based on peer-reviewed dermatological and biochemical research, not trends or commercial skincare claims. I am Dr. William Lee, and for decades I have studied how biological compounds interact with human systems at a cellular level. The focus here is not cosmetic masking, but structural repair. More specifically, how certain botanical oils, when used in a very precise sequence, may interact with deeper skin layers and influence fibroblast behavior, the cells responsible for producing collagen and maintaining skin strength. Before we get into those oils, there is a critical biological fact you need to understand. The skin on your chest is not the same as the skin on your face. That difference is not minor, and it is not cosmetic. It is structural. The skin across the chest, the décolletage region, extending from the collarbone down toward the upper sternum, is significantly thinner than facial skin. On average, it is about half the thickness. In numerical terms, facial dermis may measure around 2 millimeters. Chest dermis can be closer to 1 millimeter, or even less in older individuals. This matters because the entire support system of your skin, collagen fibers, elastin networks, and extracellular matrix components, is compressed into a much thinner layer. When that structural layer begins to degrade with age, even small losses in collagen have a disproportionately visible impact. After around the age of 60, collagen production naturally declines, and even a modest yearly reduction becomes visually amplified in thinner skin regions like the chest. There is another factor that makes this area even more vulnerable, and it is often overlooked. Facial skin contains a higher concentration of sebaceous glands, which produce sebum, natural oily substance that helps protect and hydrate the skin. Sebum is not just moisture, it acts as a biological barrier, reducing oxidative stress and helping protect against environmental damage. Chest skin, however, has far fewer of these glands. That means less natural protection, less lipid coverage, and greater exposure of the underlying dermis to damaging forces. Those damaging forces are primarily ultraviolet radiation and oxidative stress. Over decades, repeated exposure to sunlight without consistent protection leads to gradual collagen fragmentation. The skin does attempt to repair itself, but in areas like the chest, where protection is weaker and repair capacity is lower, that damage accumulates faster than it can be corrected. There is also a behavioral factor that accelerates this process. Most people are careful with their facial skincare routine. Sunscreen is applied, serums are used, and hydration is prioritized. The chest, however, is often neglected. At best, it receives leftover product from the face. At worst, it receives no consistent protection at all. This creates a long-term imbalance in skin aging between facial and chest tissue. As a result of these combined factors, thin dermal structure, reduced sebaceous protection, and chronic UV exposure, the chest develops a characteristic pattern of aging. Vertical creases form from repeated sleep compression and gravity. The skin begins to take on a crepey texture, losing its smooth elasticity. Over time, laxity becomes more visible as the skin loses its ability to rebound after movement or pressure. It is important to understand that these changes are not primarily superficial. They originate in the dermal layer, where fibroblasts are responsible for producing and maintaining collagen and elastin. In aging skin, fibroblasts do not necessarily disappear. They become less active, less responsive, and operate in an increasingly depleted biochemical environment. This is why surface-level moisturizers cannot solve the problem. They do not reach the dermis. They do not meaningfully interact with fibroblasts. They cannot restore collagen or rebuild elastin networks. Their effect is temporary hydration at the outermost layer, which can improve appearance briefly, but does not address the underlying structural decline. However, research suggests that certain botanical compounds may have properties that allow them to penetrate deeper layers of the skin and influence fibroblast activity indirectly or directly. These compounds are found in specific plant-derived oils that contain bioactive fatty acids, antioxidants, and signaling molecules capable of interacting with skin biology at a deeper level than conventional creams. The key concept here is not just application, but biological communication. When the right compounds reach the dermal environment, they may help shift fibroblasts from a dormant state toward a more active state, supporting collagen synthesis and structural repair processes. This is not instantaneous, and it is not cosmetic masking. It is a gradual biological response influenced by repeated exposure over time. And this is where the protocol begins to take shape. Not as a single product, but as a sequence of oils, each with a distinct role in addressing different aspects of skin aging. Some influence gene expression pathways associated with collagen production. Others reduce inflammatory activity that contributes to collagen breakdown. Others support cellular membrane function so that fibroblasts can properly receive biochemical signals again. Before we move into those specific oils, it is important to understand that what follows is not random layering. It is structured. Each component prepares the skin environment for the next, creating a cumulative effect that targets multiple biological mechanisms at once. In the next section, we will begin with the first of these oils and examine how it interacts with the genetic signaling pathways of skin cells and why it plays a foundational role in this entire sequence. The first oil in this sequence is not chosen for surface hydration or cosmetic softness. It is chosen because of how it interacts with cellular signaling at a much deeper biological level. This is where the approach begins to move away from conventional skincare thinking and into functional skin biology. We begin with pomegranate seed oil, extracted from the seeds of Punica granatum. At first glance, it appears like just another botanical oil, but its composition is what makes it relevant in this context. It contains a rare fatty acid profile, especially punicic acid, an omega-5 fatty acid that is not commonly found in most plant-based oils. This compound is one of the reasons it has been studied for its interaction with skin cell behavior. What makes pomegranate seed oil significant is not that it hydrates the skin. Many oils can do that. The interest comes from research suggesting that its bioactive components may influence gene expression pathways linked to structural proteins in the skin. In laboratory and pilot observations, certain compounds in pomegranate seed oil have been associated with increased expression of genes related to type I collagen and elastin production. To understand why this matters, you need to think about how skin structure is maintained. The dermis is not static tissue. It is constantly being remodeled by fibroblasts, which produce collagen fibers and elastin networks. These proteins form the structural framework that keeps skin firm, elastic, and resistant to gravitational forces. When we are young, fibroblasts are highly active. With age, their activity declines and the balance shifts toward breakdown rather than repair. Pomegranate seed oil is interesting because it appears to interact with the signaling environment that fibroblasts respond to. It does not force collagen production in a mechanical sense. Instead, it is studied for its ability to influence the expression of genes that regulate structural protein synthesis. In simple terms, it may help shift cellular behavior from a maintenance-deficient state toward a more active, repair-oriented state. One of the key areas of research involves its effect on type I collagen, the most abundant structural collagen in the dermis. This is the protein responsible for tensile strength in skin. When type I collagen breaks down faster than it is replaced, structural integrity weakens and visible wrinkling begins to form, particularly in thin skin regions like the chest. Another area of interest is elastin. While collagen provides strength, elastin provides elasticity, the ability of skin to return to its original shape after being stretched or compressed. Loss of elastin integrity is what contributes to the crepey texture and reduced snapback effect seen in aging chest skin. Some studies suggest pomegranate seed oil may influence elastin-related gene activity as well, although this remains an evolving area of research. There is also a third biological component often discussed in relation to cellular aging: telomerase activity. Telomerase is an enzyme involved in maintaining telomere length, which plays a role in cellular lifespan and replicative capacity. Some early-stage biochemical discussions suggest that compounds in pomegranate seed oil may have indirect interactions with pathways related to cellular aging markers, though this area is still under investigation and should be interpreted cautiously. What is important here is not overstating certainty, but recognizing directionality in the research. The focus is on whether certain plant-derived compounds can influence fibroblast behavior in a way that supports structural maintenance over time. In practical application, pomegranate seed oil is typically used as the first layer in the protocol. The reasoning behind this is based on molecular size and absorption characteristics. This oil is relatively light compared to many other botanical oils, which allows it to penetrate more efficiently through the upper layers of the epidermis. The goal is not to remain on the surface, but to reach deeper layers where fibroblasts reside. When applied to the chest area, the oil is introduced into a biological environment that is already compromised by thinning dermal structure and reduced natural lipid protection. In that environment, delivering bioactive compounds consistently over time becomes more relevant than isolated application. It is typically applied in small amounts, often just a few drops, to clean skin. The focus is not quantity, but consistency and distribution. The skin on the chest does not require heavy occlusion. Instead, it responds better to light, repeated application that allows absorption without overwhelming the barrier. Pomegranate seed oil does not function in isolation within this protocol. Its role is foundational. It prepares the biochemical environment, but it does not address inflammation, enzymatic breakdown, or receptor-level responsiveness on its own. That is why it is paired with other compounds later in the sequence, each targeting a different mechanism of skin aging. Before moving on, it is important to clarify a key scientific point. While studies and biochemical analyses suggest promising interactions between pomegranate seed oil and skin cell behavior, this is not equivalent to a medical treatment or guaranteed structural regeneration. Skin biology is complex. Individual responses vary based on genetics, age, sun exposure history, and overall skin health. What we can say with confidence is that fibroblasts in aging skin do not lose their potential entirely. They become less responsive, less stimulated, and less supported by their environment. The goal of compounds like pomegranate seed oil is to improve that environment in a way that may support more optimal cellular activity over time. Now that we have established the role of the first oil in this sequence, the next step is to address a different but equally important problem in chest skin aging: chronic inflammation and enzymatic collagen breakdown. This is where the second compound enters the protocol, and its role is to protect the structural gains that the first oil is attempting to support. Once the foundational signaling environment has been addressed, the next major challenge in aging chest skin is not just reduced collagen production, active collagen destruction. Even if fibroblasts are encouraged to produce new structural proteins, those gains can be rapidly undone if the surrounding environment remains inflamed and enzymatically active in a destructive way. This is where frankincense oil becomes relevant, specifically extracts derived from Boswellia species and their active compounds known as boswellic acids. Unlike the previous oil, which focuses more on cellular signaling and gene expression pathways, frankincense targets a different biological problem entirely: inflammation-driven breakdown of skin structure. To understand its role, you need to understand what is actively degrading collagen in aging skin. One of the primary drivers is a group of enzymes known as matrix metalloproteinases, or MMPs. These enzymes are naturally present in the skin and serve a normal function in tissue remodeling. However, when their activity becomes chronically elevated, most commonly due to ultraviolet exposure and persistent oxidative stress, they begin breaking down collagen faster than it can be replaced. In the chest area, this imbalance is especially pronounced. Decades of sun exposure without consistent protection create a long-term elevation in MMP activity. The result is not just slower collagen production, ongoing structural degradation happening in the background, even when the skin appears at rest. Boswellic acids have been studied for their ability to modulate inflammatory pathways that contribute to this enzymatic activity. Research in dermatological and biochemical literature suggests that these compounds may help downregulate certain inflammatory signals that drive MMP expression. In simpler terms, they may help reduce the biological signal that tells the body to continue breaking down collagen at an accelerated rate. This is important because skin repair is not just about stimulation, it is about balance. If you stimulate collagen production without controlling collagen degradation, the net structural improvement remains limited. Frankincense functions here as a regulatory compound, helping to shift the environment away from continuous breakdown and toward a more stable repair state. Another important pathway involved in chest skin aging is related to inflammatory mediators produced through the 5-lipoxygenase pathway, often abbreviated as 5-LOX. This pathway generates leukotrienes, which are signaling molecules involved in inflammatory responses. In chronically stressed or UV-damaged skin, these inflammatory mediators can contribute to tissue sensitivity, weakened extracellular matrix integrity, and increased vulnerability to mechanical stress. Why does this matter specifically for chest wrinkles? Because many of the wrinkles in this area are not only the result of intrinsic aging, but also repeated mechanical compression, especially during sleep. When inflammation is present in the tissue, the extracellular matrix becomes more fragile. This means that normal physical pressure, like side sleeping or skin folding, can create deeper and more permanent crease patterns over time. Boswellic acids are studied for their potential to inhibit components of this inflammatory cascade. By reducing 5-LOX activity, the tissue environment becomes less reactive and less prone to chronic inflammatory degradation. This does not eliminate mechanical forces, but it may reduce how severely the skin structure responds to them over time. There is also an important practical consideration when using frankincense oil in any topical protocol. Unlike lighter carrier oils, essential oils derived from resin are highly concentrated. They are not designed to be applied directly to the skin in undiluted form. This is critical for safety and skin barrier integrity. In structured application protocols, frankincense is typically diluted into a carrier oil at a low concentration before being applied to the skin. This allows the active compounds to be delivered without overwhelming or irritating the skin barrier, which is already compromised in aging chest tissue. Within this multi-oil approach, frankincense serves a stabilizing function. If pomegranate seed oil is focused on encouraging fibroblast activity and collagen signaling, frankincense is focused on protecting the environment in which that collagen is meant to form. It reduces the biological noise of inflammation and enzymatic breakdown, allowing repair processes to proceed with less interference. However, even with reduced inflammation, there is still another problem that must be addressed. Fibroblasts in aging skin do not only suffer from inflammatory stress, they also become less responsive to key signaling pathways that regulate collagen production. In other words, even when the right signals are present, the cells do not respond as effectively as they once did. This brings us to the next compound in the sequence, which focuses on restoring cellular responsiveness to those signals. It works at a different level entirely, targeting receptor pathways that influence how skin cells interpret regenerative cues. Even when inflammation is reduced and collagen breakdown is slowed, there is still another major limitation in aging chest skin that often goes dressed. The problem is not only what is happening outside the cells, but how the cells themselves respond to repair signals in the first place. Fibroblasts, the cells responsible for producing collagen and elastin, do not simply turn off with age. Instead, they become less responsive to the signaling pathways that once kept skin structure stable and resilient. This reduced sensitivity means that even when regenerative signals are present in the environment, the cells do not react with the same efficiency or intensity as they once did. This is where bakuchiol enters the protocol, a plant-derived compound extracted from the seeds and leaves of Ceralia coralifolia. At a structural level, bakuchiol is often discussed in relation to retinoid-like activity, but what matters most in this context is not branding or comparison, but its effect on cellular communication pathways in skin tissue. Retinoids such as tretinoin are well known for their ability to influence gene expression in fibroblasts, increasing collagen production, accelerating cell turnover, and improving dermal thickness over time. However, they also come with a well-documented downside: irritation, barrier disruption, and increased sensitivity, particularly in thin or already compromised skin, such as the chest. Bakuchiol is studied because it appears to interact with similar receptor pathways associated with retinoid activity, but without producing the same level of inflammatory irritation. In controlled comparative studies, both retinol and bakuchiol have shown improvements in fine lines, pigmentation, elasticity, and overall photo damage. The key distinction lies in tolerability, with bakuchiol generally demonstrating a lower incidence of irritation-related side effects. In the context of chest skin, this difference is critical. The skin in this area is already thinner, more exposed, and structurally weakened compared to facial skin. Introducing a compound that triggers irritation or barrier disruption can paradoxically accelerate visible aging by increasing inflammation and compromising the skin's protective function. Bakuchiol, by contrast, is used in this protocol for its ability to stimulate cellular activity in a gentler, more stable way. The focus is on encouraging fibroblasts to behave more like they did in younger tissue, where response to regenerative signals was stronger and more consistent. At the biological level, this means supporting pathways involved in collagen gene expression and cell turnover without pushing the skin into an inflammatory stress response. The goal is not aggressive stimulation, but sustained reactivation of natural repair mechanisms that have become less efficient over time. This is particularly important when considering how multiple compounds in this sequence interact. While pomegranate seed oil is associated with signaling that may influence collagen gene expression, frankincense helps reduce inflammatory degradation. Bakuchiol acts more like an amplifier of responsiveness. It helps ensure that when fibroblasts receive regenerative cues, they are more likely to respond in a meaningful way. In that sense, bakuchiol does not replace the earlier compounds. It enhances their effectiveness. It helps bridge the gap between signaling and response, ensuring that the biochemical instructions delivered by other oils are not only present, but actually acted upon by the cells. Another important aspect of bakuchiol is its influence on skin turnover. As skin ages, The natural process of shedding and replacing surface cells slows down. This contributes to dullness, uneven texture, and a buildup of older, less functional cells in the epidermis. By supporting a more balanced turnover cycle, bakuchiol helps improve the overall environment in which dermal repair occurs. However, even with improved cellular responsiveness, there remains a structural limitation that must be addressed at the membrane level. Fibroblasts rely not only on internal signaling pathways, but also on the integrity of their cell membranes to receive and transmit biochemical information effectively. As skin ages, the lipid composition of these membranes changes. They become less fluid, less responsive, and less efficient at transmitting signals from the external environment into the cell's internal machinery. This is one of the subtle but significant reasons why regenerative signals become less effective over time, even when they are still present. To address this final layer of dysfunction, the next component in the protocol focuses on restoring membrane integrity and improving cellular communication efficiency. It targets the lipid environment that surrounds fibroblasts, ensuring that signals generated by the previous compounds can actually be received, processed, and translated into structural repair activity.